Hello, everyone. Welcome back to the Bioinformatics Workshop. Today, I'm going to go over Introduction to Genomics and Viral Data Analysis. And we're going to use the things we learned from the first two workshops. And today, we will actually do some bioinformatics. If you have any questions along the line, like along the workshop, feel free to go to slido.com and use the participant code 3661069. And Rose and Ferial will monitor your questions. As well, for online, for in-person participants, feel free to raise your hand and we will get to your questions. Just a reminder that this is our schedule and we are at the third week, March 16th. Next week will be next 4 weeks will be all bacteria data analysis. So if you're a bacteriologist and would like to learn how to do data analysis, then feel free to attend these sessions. And for in-person participants, if you're new, please connect yourself to U of M Secure if you're a student or staff. If you're a guest, you can temporarily use the U of M guest Wi-Fi. Today's learning objective is as follows. So last week we did conda installation and tool package installation, but we had a problem where Jill actually found out that SAMTools was not properly installing, and she figured out a whole method to actually get it to properly working. So I will go over this today briefly, and then we will update our package so it works when we need to use it. And then the next, we will go over just type of viral genomics data analysis. So how some people are using it. I only do it genomic surveillance, so I only know one sort of method, but there are other ways how genomics data can be applied for research. And I'll provide a background on Nanopore long-read sequencing because today's data that I'm using is actually from King al. paper, and they used long-read Nanopore sequencing. So we're going to work on the bioinformatics data set and using the tools to analyze this data. And then briefly go over the fastq data analysis steps that I will use today, and we can actually do hands-on in our last objective. So if you attended last week's workshop, or if you watched online, like a YouTube, you probably got to installing the Miniconda 3, and yeah, you probably didn't realize if you didn't install SAMTools, because SAMTools was the one that had problem. So for newly installed Miniconda 3, anybody who newly installed for this workshop purpose, your channels need to be configured if it's your first time. So if you had problems, it's with certain packages because you probably only saw default channel on your conda in the conda environment. We actually need to have bioconda and conda forge configured to your channel as well. So Jill had this whole figured out, and I will go over the code today. So first, you can open your terminal and check your conda channels. So you'll type conda config -- show channels. Okay, so if you open up your Ubuntu terminal, you should see a bracket base. Okay, so we're going to type in this code. Now, if you don't see 3 channels and you only see defaults, you need to configure your channel, conda channels. To do that, we're going to add the following channels in this order if you don't have it configured already. So I can just show you. So you do conda config -- add channels. And the first channel you have to add is conda forge. I'm getting this warning because I already have the channel here. But you should just have, it should just work for you if you didn't have this channel. And then the next, we're gonna add bioconda. So once you've added your channels, we're going to check the conda channels again. And you should see 3 channels in your terminal. Once you have these following channels in this specific order, so bioconda has to be at the top because it is the most important one. And then conda forge, which has all dependencies. And the default is Anaconda basic channel. Then you can update your conda package, like the tools that you've installed for last workshop. So if you're having trouble with SAM tools, we can update that. So we'll do conda update -y for yes, -c to specify bioconda channel and the package name. So if you press enter, oh, sorry, before you do this, you actually have to you have to activate your conda environment. So we're gonna do conda activate conda workshop. Because we've installed these packages in our conda environment that we've activated. So we have to activate our channel first. Make sure you see conda_workshop inside the brackets. And then now you can run the conda update -y-c bioconda samples. Okay, it looks like it's working. Oh, if you haven't installed your package, if you had trouble installing, what you can do is you can do conda install -y-c. Oh, yeah, maybe do this first. Conda update if they for sure know. So some people who are having trouble that there is package not installed, like error message. If you've installed the program, like the package, and you know it, you can do conda update -y and just SAM tools. Maybe give that a try. And if this doesn't work, then your package is not installed. So you can just reinstall the package. So if this didn't work, the command to install package is conda install -y -c bioconda SAM tools. So if you did the install conda install SAM tools, maybe try doing the update. So if you've configured all the channels and you've updated, then you should now see specific channels for each package. Because before we've only seen Bioconda for the package that we installed. Everything else was empty, but now you should be able to see Conda Forge beside the dependencies and so on, and Bioconda for the ones that you've installed. In case you've added the Conda channel in a wrong order, you're seeing like Conda Forge at the top instead of Bioconda, then you can also remove your channels and then re-add the channels. Now for this workshop, there are several tools that I asked, or last workshop I asked you guys to maybe try it at home and then install. So we will go over the tool package that we need for today's workshop, like how to install it, and also update the previous packages. So the command to see the conda list again is conda list. So you want to see your package that you've installed and the list of channels where it is built from. So it's conda space list. So we will first install the conda package for today's workshop, which is Nanoplot. So you've already activated your conda_workshop. Then from there, we will directly install conda Nanoplot. Make sure your base is turned into conda workshop if you've activated your environment. Conda install, yes, channel name, Bioconda, Nanoplot. And if you want to know if the package is available, you can do Nanoplot or specific package search up on conda. A very simple way to check. And if you have it on the Anaconda website, then this is available as a conda package. So install nanoplot. I already have these tools installed, so your screen and my screen might look different. Once you got your prompt back, we will move on to now updating our package that we installed previously. For time's sake, for today, we will just update the Minimap 2. If you've already installed these package, we'll just update Minimap 2 and SAM tools because that's the tools that we are going to use today. And the rest, you can try it at home or on your own time to update, but make sure to have them updated before next week's workshop. So for this one, instead of specifying the channel, if you've already installed, I found that it didn't really work when I did the -c bioconda. So here we're just going to do conda update -y and Minimap 2. And once again, if you haven't installed these package, you can simply use conda install -y-c bioconda and the package name. We've already done conda update for SAM tools, so we don't have to do it. But if you haven't done it, you can do the same conda update -y SAM tools. Okay, so now we will move on to the next step. So now that we've configured our channel and have content tools ready, we will go into data analysis aspects of genomics data. So I will briefly explain some common methods that we use or labs may use for viral genomics data analysis. So why would the lab be one to sequence virus genome? Genome sequence or any genome sequence can be used for detecting drug resistance, understanding the virus pathogenicity, but this also can apply to bacteria genome or fungal genome or barley, like if you're in agriculture, and also for virus genomic surveillance. For the past two years, we were in COVID-19 pandemic. That we had a COVID-19 pandemic, and part of the project that I worked on was doing the whole genome sequencing of SARS-CoV-2 virus to get the lineage profile. And this helped us understand epidemiological link, the cases, maybe like one outbreak occurring from this location. Do they have the same type of lineage profile or do they have different one? Is it an emerging lineage or like Omicron, Delta? they were kind of sort of new in certain aspects of the genome. Like, so they were emerging variants of concerns, but other viruses, other strains, or like the lineage profile might be same, then they're the same thing circulating. So those are some type of questions that you can potentially answer, maybe not fully understand, but at least get an idea. And that's when the genome sequence data comes in handy. These are just few examples, but there are certainly many applications why one would be interested in sequencing genome. So like, for example, a drug resistance that I'm kind of aware of from like colleagues working on HIV, they would be sequencing the virus to determine their like genome profile and see which part of the genome is susceptible or it's not susceptible. It's resistant to antiretroviral therapy. That's one way how sequencing can be used. And here is my example, genomic surveillance to determine the phylogeny and the relationship of the circulating SARS-CoV-2 viruses. So for today's workshop, we are going over King et al.'s paper, and they use Nanopore long-read sequencing. So I'm just going to go over some technologies available for genome sequencing. So there are 4 that I'm sort of like well aware. Certainly there are other tools available. The Sanger was the gold standard for many years. It's not really capable of doing complete like whole genome sequencing data, but it's very accurate. So Sanger sequencing was used for a while until the next generation sequencing came along. Illumina sequencing is very accurate, but it's good for short read sequencing. It's more targeted towards that. And then eventually Nanopore sequencing technology was introduced, and this is targeting long read sequencing like PacBio. Yeah, so these are 2 long read sequencing technologies. And from there, I have more experience with Nanopore. I've never had an experience with PacBio, and I have very little experience with Illumina. So that's why the data is geared towards Nanopore. I'm a bit biased to Nanopore sequencing data. So we will use that data set to analyze data. So typically the workflow of genome sequencing would be like, you'll get clinical samples from patients, or you can get environmental samples, whichever, and you can do targeted PCR. So like amplifying the specific genome specific region of the gene. Or you can do a whole genome sequencing where you do untargeted PCR, or maybe just untargeted PCR. Just one method, and then you can put it into a sequencing machine. So this is like Illumina, but you can replace that with Nanopore. You get sequencing, and then the output is used to analyze data. And this is where the bioinformatics come in for the next generation sequencing data and the Nanopore and PacBio. It's more bioinformatics heavy, so we will do this part of the workshop today. For this workshop today, we will do this part of the analysis. So Nanopore sequencing technology is kind of unique that it has a nanopore embedded in a plate where electricity passes through. And as the DNA goes through the nanopore, it generates a weird signal through by disrupting the ionic channel, by disrupting the electricity, and that leads to generating a specific signal for a specific nucleotide. And so this is really the beginning of the base bioinformatics part for nanopore sequencing data, because we get an output as fastq, and this is the signal data. And then this has to be converted into fastq. And now the sequencer has mostly embedded this base calling step as kind of like a part of the sequencing machine. But before we had to separately do base calling after we get the fastq output to get the fastq, run the read mapping or de novo analysis, and get the consensus genome. So for today's workshop, the King AL paper, actually, it's an influenza A virus sequencing paper. So the authors developed a high throughput sequencing workflow and a speedy screening method for unknown IAV samples. Now, their data analysis method includes Guppy. Guppy is a tool for base calling of nanopore reads from fastq to fastq. And then they do run demultiplexing because you can sequence multiple samples at once by having separate unique barcode. So that barcodes can be separated by demultiplexing. So barcode 1 would be sample number 1, barcode 2 would be sample 2, and so on. And then part of the tool includes the adapter trimming automatically. And then third step would be nanoplot to generate read statistics. And then their last step was using Genius. In this case, they use Bowtie2 read mapper to map it against a reference. This paper was actually comparing their results with another sequencer. So that's why the analysis method may differ depending on what your experience is, but we will only focus on the nanopore aspect. So for our workflow today, I'm going to modify it a bit because we've already got the fastq data. So I've uploaded just 2 fastq files, but if you are interested in practicing, you can go to the paper cited. And the paper is also available on the GitHub page, and download more files. From fastq, because it's already base called and demultiplex, we're going to go straight into doing nanoplot, read statistical generating the read statistics, run minimap2. So instead of Bowtie2, minimap2 is more geared toward long read sequencing data for like nanopore. So we will use minimap2 to read map against a reference. And then use SAM tools to convert the SAM output from minimap2 to BAM. And then view the BAM files on IGV. It's an integrated genomics viewer. It's a separate software that will show us the read coverage and the nucleotide mutations that may have been found that's different from the reference file. So here's just an idea of reference mapping. So we get a reference fast genome, and then we map our reads against the reference to get the information and eventually generate a consensus of like the genome that we've sequenced. And this can be used to identify the virus, or you can check which type of mutations are present that was not present in the previous few months ago, or maybe many years ago. Okay, so now we'll go back to doing our hands-on. So I will show you this through my terminal, but these slides are available on the GitHub if you'd like to follow along with the slide on your computer. Okay, so first thing first, you're gonna open your terminal and change your directory to the preferred destination. So for this workshop, I like to keep everything on my desktop, but if you have a different location, you wanna put it in documents folder or your downloads folder, feel free to change your directory into one of those. So I'm going into my desktop. And if you want to check the contents of your desktop or your folder, you can do L for vertically listing all the files, or you can do ls, and it shows you only the file and folder name. So once you're in one of the directory that you want to be in, you're gonna do git clone and this git website. So if you remember from last week, if you go to our website or go to the Eventbrite email that I sent yesterday, you'll have a link to the workshop repository. So once you get to the repository, you can click code. And under HTTPS, here is a link. You can click to copy this URL. Or you can simply do download zip. But I think it's faster if you do it on command line. So just copy the URL and do git clone. HTTPS. If you don't have Git, you can actually install Git by doing sudo apt get git, I believe. I already have this file from testing the folder, so it's telling me that I can't do another git clone. So once your cloning has worked, then now you can change your directory into the cloned Git repository. So what you want to do is so you can tab complete after typing certain number of characters. If it's unique, you can do a tab complete so you don't have to type the whole thing. So you'll have to do CD 2023 intro to that folder that you just copied. And if you list the contents, then you'll see the folders King and VWigger. So the same folder you should see on this repository in GitHub. From there, we're going to go into King AL 2020 folder. So CD King AL 2020 folder. And if you list your contents again, dataset is inside data_influenza. So you want to go into that folder. Now you should have 2 fastq gzipped fastq files. Once you've gotten this point, now we're going to create directory to run our analysis and save our output. So what I always like to do is for my analysis folder, when I make one, I like to include the date and the time I ran the analysis. So to do that, you can make a variable to hold the date information and use that to make a folder, or we can simply do this. So the date variable, so in this empty object, I'm going to make sure that I get this date command. So if I show you the date command automatically populates today's date and time. So from there, I want a specific format. So if I want specific format, you can do man page on date command. This is what I like. So I want the years to come first, 2023. And then the month. So you have plus sign, percentage, capital Y to list full 2023. And then the percentage, M, lowercase, to specify the month, and then percent D to specify date, put underscore, percent capital H for hours, and then percent capital M for minutes. So if you type this command into your console, you're going to get the following output: 2023 March 16th underscore 0508. Well, not 0508, depends on your time zone. Okay, so this information, I am putting it into my variable by typing the word date equals dollar bracket, put in this date command that we've just got, and then close it with the bracket. Now, if your command worked, you can check your variable echo $date. Anything variable, if you want to call the variable object, you have to put $sign in front or money sign in front of it. So if you do that, then you've got your variable. Or you can manually input this. So what we're going to do is we're going to now create in your, so we're going to go back to our King AL directory, get out from your data influenza. So CD up a parent folder. So your current directory is now King AL 2020 folder. So from here, we're going to create new analysis folder called analysis underscore and put $sign if you want to call your date variable. And check if you've made the folder. So it worked. So we're going to go into your analysis underscore date folder now. So your new current working directory should be that analysis folder you just created. And there shouldn't be anything now in there. So now that you're in your analysis folder, we're going to create a folder called nanoplot. This is where we're going to store our nanoplot results. I like to put numbers in front if I do multiple types of data analysis, but this is totally up to you. You can just call it nanoplot or whichever naming scheme you'd like to use. I just like to keep the order of the analysis consistent, so I use numbering system. Once you made the nanoplot folder, go into the nanoplot folder then. Now your working directory should be quite long, King AL, analysis, and then nanoplot. So now here is where we're going to make directory again, but for specific samples. So we have two fastq files. We're going to make folder for sample number 1 and then sample number 2. You can actually you can use mk dir-p, multiple folder name, just add space between, then you'll make 2 or 3 or how many ever folder you are generating. So in this case, I'm doing 2. I'm just saving myself a line, typing both sample 1 and sample 2 folder at once. So let's check. Right now, there's nothing in here. But if I do, two folders together with the space between the name, I've created 2 separate folders. Okay, so just going over the slides. These are all in there. Now we are going to run nanoplot. So just to note that this NN sign you see in these previous folder here, I make directory, and then I append the next command. Once directory is made, go into the directory. So this is what this double N means, the symbol. I just did it step by step when I'm showing it, but in the slides, it's actually doing multiple things at once just to save a line. It's exactly the same as what I just showed. Okay, so now you have this folders generated. We're going into, let's try sample number 1. So change your directory, go into the directory 3822171. One way to make sure that you are in that directory is you can actually see where you are. If you have a full path listed, or if you have shortened version, the last one the first one or last one you can see is the current directory you are in. So once you're in there, now we're going to run nanoplot. Nanoplot. So I'm just kind of using the default command that's available publicly that many people use. You can certainly optimize by looking at the parameters. We're just going to do this. So -t stands for threads. We're going to use 2 threads. And we're going to specify our fastq files. So in this case, we are running nanoplot on sample number 1. So we have to specify the path to this fastq file by doing double dot. So going up a parent directory, but we have to go up another one because we have to get into the analysis folder. So by doing this, we're currently here. We go up here, nanoplot. That's the first double dots. In the second double dot, we're going into the analysis folder. Oh, yeah. So, sorry. So the first one, we're going from this to the nanoplot folder. Second one from nanoplot to the analysis folder. And then third double dot to go into the King AL folder. And this is by using relative path. So from where I am, I'm going into data influenza folder. So how can you check if you actually is doing the right input? You can you can actually tab complete, see if you can get the contents. So like I say, I write data. If I'm currently in the right location, then I should be able to tab complete to get that data underscore influenza automatically filled out. And then I'm going to do ERR, tab. And now 1 or 2 is not unique. There's unique ones. Computer doesn't know which one I want. So I just have to fill out number 1 and then do tab complete. Specify my fastq. And then the maximum length, 4,000. We don't, you can specify it if your sequence data is long, like maybe 8 KB, you can go further, like higher, but I know for this one, it's not that long. So we're gonna just say maximum length, 4,000. If anything beyond that will not be part of the nanoplot output. And then give it a space, --tsv stats. We want the output in TSV format, tab delimited. And then you can run it. So this shows that computer is processing my request. There's no prompt. So you can't do anything for now. We'll just wait until it outputs the results, or once we get our prompt back. So now we got our prompt back. We can check if it actually ran properly. We will do L. From nothing, you should now see these list of files generated. Let's go to the folder for people who are familiar. So now you should have nanoplot folder. We ran the first sample. It's showing HTML or an image file. You can check yield by length, nanoplot report. This is a overall report summarizing all of the files here into one HTML. So if you open nanoplot reports, you should see the following output. How many reads were there? How many bases? What's the read length? And for Nanopore, the quality score is the default standard is 7 or above. So it's showing you how many reads were above quality score of 5. So that's 100%. That's good. And then how many reads were above 7%, Q7, 99.9%, and so on. You can see all the list. The higher the Q score, the better the sequencing results were. So if you have reads that are higher in quality score, then that's kind of similar to Illumina, where you want the higher quality reads. No, so this is for Nanoplot is specific for more like the Nanopore reads sequence. And you can get an overview of graph, and you get the nice output generated. And these are interactive, so you can actually see X and Y if you hover over. So this is just a quality report you can get from Nanoplot. You can get the similar thing from fastq, but personally, I haven't really used it much for Nanopore data. So if you're attending Jill's next week workshop, she will be going over that in detail. So great, now we've got the output for the first sample. You can repeat the same for the next sample. The same thing, but instead, now remember we have to go back a parent folder because we are currently in sample number 1's folder. We're going into this list of folders again. And from there, to run the analysis for the second one, we're going to do ERR CD into this folder and repeat. So I will leave the second one for you to try it on your own, and I will go into the next one. So I have the code available for both sample 1 and 2, but the sample number 2, I will let you try it on your own time. Okay, now we're going into the Minimap2 data analysis. So if you need the plot, if you need the code, the slides have the codes already for the second one as well. So Minimap2 is our reference read mapping tool that we're going to use. So for this one, we need to, before we can use the reference file, it is better if we create an index for your FASTA file. So what we're doing is we're going to create an index, reference index file in where we have the reference file available. So I put the reference file that's influenza, that's reference underscore influenza, that FASTA. It's available in the King AL folder. So the path, just whichever path you've used, you can use that to go into the original. So somebody was asking how long Nanoplot would take to process. It depends on your processing power, yes, definitely. So, but if you have about 16 gigs of RAM and i7 core, it should be running like within a few minutes. But it also depends on how big your sample is, like your fastq. If it's a gigantic one, sometimes even on the cluster like supercomputer, it takes it could take up to a few hours. So that just depends on your sample. For this one, it shouldn't take that long because the sample is small, like the fastq file is small. Should take about a few minutes or maybe longest 10+ minutes. So from our current folder, we're going to King AL folder. So you can use relative path. And if you double tap while you didn't press enter, but if you double tap, you can actually see the contents. It kind of helps you. Oh, maybe I went too far, or maybe I didn't. So I have to go back. Then now I can do King AL type, or in this case, I went too far. So I'll just do double dot, or sorry, go up to parent folder, and you should see influenza underscore references.fasta file. So this MMI is Minimap2 index file. It's already made because I already ran this analysis, but if you should only have influenza underscore references.fasta. So for you can also use absolute path, depending on which path you've used up front, and then go into the King folder. So to run this command to generate minimap index for our reference file, we're going to do minimap2 -x. X is an accessory, it's a special option available for ONT. So it's, if you've done Nanopore, there is already default parameter available for you to just use it. So -d, specify what your .mmi output will be. And then what your influenza reference file is. So this is my original file that I have, and I'm creating an output for .mmi. And it should take like a few seconds. So if you've created this index, you should get this output right away. And then see this .mmi file in your folder. Now we're going into this analysis folder. since you only have one analysis folder, you should just be able to tab complete, or you can do cd analysis underscore, use a variable $date. Now, once again, if you've changed your environment or like your directory, you should see that in your prompt. And now in the same folder, we have nanoplot folder. Now we're going to create minimap2 folder. And then go into that minimap2 folder once you've created. And you should see you're now in minimap2 folder. Now you're going to run minimap2 on the fastq file. So again, the same fastq file you're using. So we'll do minimap2 -ax. So if you do minimap2 -h for help, you should see the list of presets and file options parameters available. What I'm doing is very basic. So if you want something more specific geared towards your data, you can look through these parameters and improve your output by doing that. So we'll do minimap2, use a preset, map ONT, and we want to use influenza references FASTA. So we're going to specify our reference file first. So reference file is, I compiled it for you, just a few influenza files, but you can always create it just using a notepad. And then specify that output as .fasta. If you know what you're looking for. Okay, so I have this influenza reference FASTA file. And then now I'm going to specify my fastq file. And I'm doing number 1. So two folder up, and then go into data influenza, specify this fastq file, number 1, and then save that output, reference mapping output into SAM. So Minimap does SAM output. So we are going to use another tool to convert that into BAM. So if you run this, it shouldn't take that long either. So again, try number 2 on your own time, but the command is there for you. Okay, so it only took 12 seconds to map the reads. Yay. So now we are going to convert this output. So we have the SAM output file. We're going to convert this into BAM using SAM tools. So BAM is a binary file, so that's why but it's easier for programs like IGV to process. They only process binary file. It's faster for the computer to process with an index. That's why we are changing it into BAM output. So to do that, you have to use samtools command, samtools -vu -s-p. Specify that SAM tool in your current folder, you have that SAM output. We're just going to change the extension into that BAM. Oh, so if you get an error like that, it failed to open, no such file or directory, I definitely made an error in my. Yes, I forgot to add 7 here, so that's why it didn't work. So we're just going to remove that BAM. I mean, it's empty, it should be okay, but I just like to remove it. So to remove a file, in case of a mistake, you can do RM. So now let's repeat this again. Just make sure you do SAM tools view SAM into BAM output. Oh my gosh. I did the same thing again. Okay, now it should work. My input was wrong, not my output. So hopefully it should work now. Yes, it's working because I see that prompt is kind of held. So while that's running, yeah, so once you get your prompt back, you've got your output. So now you have the SAM file and BAM file. And notice that how smaller it got from the SAM output to BAM, because binary file is more efficient for a computer to process. It reduces the file size. Now that we have this BAM file, we have to sort it so that when we build an index, computer can find, like our software can find the reads much faster. Another level of improving the efficiency. So to do that, we will still use SAM tools, but we're going to use sort. But now we only concern about BAM files. So we're going to do our input as this BAM output. I'm going to add the word BAM, sorted BAM. So I know that I want to use a sorted file. So once you're done running it, the last step is really creating an index file so that IGV can open it. So we've generated from SAM to BAM to sorted BAM. And we use sorted BAM file to generate an index. So the final step is SAM tools index sorted BAM file as an input, and it will create .bai, I for indicating that it's an index. Similar to MMI from Minimap2, MMI, I stands for index in extension. So you've created two files. For IGV, you need to have a VAI file, like an index file available, otherwise it will not open. Okay, so now that we've got through, so all of these commands are available in here. The second one, try it on your own time. And then we've got the sorted, and then generate an output for index. Now we're going to visualize our BAM file in IGV. Now you may be familiar, you might not be. So I've included the IGV download in the README file on our GitHub. So I'll just show you what IGV looks like. So IGV, first thing you have to load is you have to load the genome from file, and this is your reference file, that FASTA extension. So you, under the genomes, click load genome from file. And then you put in your FASTA that you got from this GitHub folder. And then it will now show up as influenza references.fasta. This tells you that this is the reference you're using to view your current file. Now, if you go under file, you can now load from file, load your BAM files. You have to load your reference file first in order to load your BAM file. So once you open up the file, you can select the files. You want to click sorted BAM, but make sure you have BAI file available for each sample. And they have to have the same name, just .bai. You can't have a different name for your BAI. It will not be recognized by the program. So once you click on it, Now you can load it up. So because there's 4 different references that I included in this file, like reference file, it will not show up under all, but you can specifically click on influenza reference file you want to see and compare. Once you do that, now you'll see the reads mapped and coverage and the nucleotide mutations that might be present in above 20% or 15% threshold that you've set on IGV. If you choose another reference file, you notice that it barely mapped anything. So this is one way how the BAM file gets used. So now that we've generated this BAM file, I'll show you how it's done. So you can install IGV for your operating system. You can do that when you have some time. So by default, it will have some kind of like human reference file, but that's not what we want because that's not what we mapped our reads against. So we're going to do under genomes, load genome from file, go to your King AL folder, click on the influenza references FASTA. And also if there's an index file available. IGV will also generate an index file automatically for FASTA. It's small enough that it can do it automatically. Now you've clicked on it and you've generated, you now have this on your reference. Now under file, now you're going to load your BAM file. Load from file. From here, you go to your analysis folder, click your minimap 2, and click sorted BAM. Now, once you've loaded that up, it looks like there's nothing. So we have to specify reference file. If I go like this, well, there's like barely anything here with this reference. But if I go to DQ, I have a giant, like a lot of read coverage for this. So this sample is most likely this reference influenza virus. And if you want to populate, like if you want to take a look at it in details, it's 2 KB. If you go, you can click zoom in and out and see the ratio by hovering over. You can do hover over, you can click on it. Either way, it will show you how many reads are like have nucleotide A versus C versus G or T. And which reference, the reference sequence has C. So usually IGV populates colors for the ones that may be different than your reference file in a certain percentage threshold. So this is how you would kind of see results in your sequencing results in IGV, kind of get an idea. And for our purposes to get the consensus, IGV is kind of nice if you're more like if you're not ready to do complete bioinformatics on terminal, you can right click on one of these reads and then copy consensus sequence. Then open Notepad and save the consensus sequence. And you can blast this going into the website, blast this to see which region it actually maps to and which virus the part of the genome it maps to. So this is one way how some people analyze their virus data. So that's really what I wanted to show you today, just like a very simple analysis method for sequence data you may have. There's there's better way. There's different way depending on what questions you're answering. But this is just one simple way I wanted to kind of show you guys. So we've reached the end of our workshop, got all the analysis, and just do a practice on your own. So you get familiar with just running the tools on conda and bash terminal. And be ready for next week's bacteria data analysis if you are into bacteria data, into bacteriology. So for a sequence file and the file formats, different tools, they always have some kind of guide or manual on GitHub when the programmer first develops the program, they would most likely try to put manual so people can use it more easily. So these are just some of their helpful resources for today's workshop relevant, but there's more, honestly. So if you find something more interesting, more useful, that's definitely the better guide. Feel free to use those. And with that, I will conclude today's workshop. So thank you for attending. And we've moved the exit survey to Google form. Thank you, Rose. So if you have some time, maybe today, before you leave the workshop, here is a link to our Google form. We've greatly appreciate your feedback so I can improve teaching, and also our next instructors can kind of get an idea of how to maybe change their workshop or maybe modify it a bit. So anyways, that's it. Thank you for attending, and let me know if you have any questions. Otherwise, see you next week.